covered some of what's in this chapter in A&P 1. So he said you talked about the Krebs cycle, right? The citric acid cycle, probably in more detail than I would have. Did you talk about, and you talked about oxidative phosphorylation, what happens inside the mitochondria? Good, and we can skip some of those things today and spend some more time on some other stuff. All right, so onward. So I'm just going to blaze through some of these. Citric acid cycle, you've heard about it. This is my short version, right? So I am not going to test you on all of the chemical details of this system because, well, to be honest, I had trouble remembering all that stuff after all this time. But this is the short version, and this is what comes up clinically more often than not. So that's my summary. Okay, so that hopefully will fit right in to what you learned. If it doesn't, please let me know. Okay, so that. Oxidative phosphorylation also has a summary. I think in concepts more than like chemical equations, so that's kind of how I have addressed it, and that's how it'll show up on the test, right, is what moves around, not necessarily which bond changes from this to that. Okay. And we talked about this last time, but I'll start here today. Okay, so the one way of organizing metabolism, which is a big complicated topic, is to take the chemicals involved and sort of layer them out by complexity. So the nutrient pool, these are the building block molecules right here. So every part of you is made of these things, and so these building block molecules get built up, that's anabolism, into more complicated structures like triglycerides, glycogen, proteins. And these nutrients can also be broken down further and fed into the mitochondria to create ATP. Right, so we talked about that yesterday. And even though, this to us is biology, all living things on Earth share a lot of these metabolic pathways. The truth is, the fact that this system evolved like this is really pretty remarkable. That we have this common set of building block molecules that living things from single-cell organisms to huge trees to giant whales all use these same building blocks to build different things and therefore be different. So it's kind of a cool system that evolved on Earth to create all the diversity that we all appreciate so much. Okay, so the nutrient pool can go up or it can go down, and if we're going to burn it, that happens in the mitochondria, right? Citric acid cycle, and the oxidative phosphorylation is also called the electron transport system. Hopefully that will sound familiar to you all. Okay. So where do the nutrients come from? Well, this is what we were just talking about in this last week. The principal source of nutrients in the nutrient pool right here is, of course, the digestive tract. We ingest nutrients in the food that we eat. That material gets broken down into those building block molecules. More on that later in this chapter. And they get absorbed, and then they appear in the blood so that the cells can use those nutrients coming in from the blood. But pretty much all animals have pretty robust systems for, well, what happens when nutrients are not coming in through the digestive tract? This happens. In most of us Americans, this happens for at least a few hours every day. And that's when in the early hours of the morning when we've been asleep and we haven't had anything to eat for 4 or 5 hours, we get into the case where there aren't any nutrients coming in anymore. So what does the body do when that is the case? And of course the severe form of that is starvation, where there's no food available in the environment for an organism. So the nutrient input goes way down. Now, how does the organism survive that challenge, that metabolic challenge? Okay. So we have 2 sites for nutrient storage. The liver stores nutrients as kind of a fast recharge, fast discharge battery. Okay. The liver can store about 6 to 12 hours worth of nutrients for the body, just in the liver alone, okay? And we'll talk about how that storage happens here in a minute. But the liver works like that. But then we also have long-term storage of energy too, and that's found in the adipose tissue. Okay? Fats store a great deal of energy in a very small space. So because of that, when the body has excess calories, instead of just burning them as heat, let's say, that chemical energy is stored in triglycerides. And triglycerides are essentially 3 fatty acids that are on a glycerin backbone. Okay, you're going to hear me talk about this word, so let me just draw this molecule here. Well, you all are in organic chemistry, most of you, so this will be very simplistic. But we have 3 fatty acids. You remember that this, right, is carbons and hydrogens. Okay? The thing about fats is there's very little oxygen in these molecules. And carbon-carbon bonds are they store a lot of energy compared to like a carbon-oxygen bond does. So these long chains of carbons and hydrogens have a great deal of energy stored in them. And then this molecule right here, this piece, is glycerol. So we call this a 1, 2, 3, a triglyceride, because it's 3 fatty acids, so this is a fatty acid, attached to a glycerol molecule to make a triglyceride. And this is our body's principal fat. So when we have excess energy to store, triglyceride is how it's stored. First the fatty acids are made, then they're attached to a glycerol backbone, and we have this very stable fat. And it's not just human. This is animal fat. Animal fat is typically triglyceride. And why is it different? Because it stores a little more compactly, and its melting point is much lower, so it stays liquid. So that's what we find in the adipose tissue. Adipose cells look a little bit like this. They have little tiny nuclei and great big fat droplets inside. So when there's excess nutrients, that gets made. When there is a nutrient deficiency, that fat is then broken down to return nutrients to the nutrient pool. Because fatty acids can be transformed into carbohydrates, for the brain in particular. Fatty acids can be used directly by cells to produce ATP, which we're going to talk more about later on. And fatty acids in the liver can be transformed into quite a few of the different amino acids. Not all of them, but quite a few. And that last piece is our weakest link, so to speak. We can't turn fat into protein very well. Okay? So we have this big energy, this nutrient storage in the adipose tissue. Skeletal muscle itself also serves as a kind of nutrient store, but it isn't a very good one because skeletal muscle is useful in other ways. Adipose tissue helps to keep us warm, but that's its only other physiologic function. Skeletal muscle actually moves us around, right? So we don't want to break down muscle to take the nutrients out of it unless we are starving, okay? So there is a ton of protein locked up in muscle, but unless you are in protein starvation, you will not release that protein for use by the rest of the body because skeletal muscle is good for other things. So at the tail end of a starvation scenario, you will start to see a reduction in muscle mass. And that's how you know that you've sort of flipped that edge and muscle is starting to be broken down to keep the body alive, right? So it's a last-ditch effort, using muscle to provide nutrients for the rest of the body. But in a terrible case, that's exactly what will happen. Okay, so that's muscle. So why? Why do we need nutrients in here all the time? Well, all of our cells have needs, right? They all need to create ATP. They all need to do anabolism to repair and grow and maintain themselves. And then our neural tissue is particularly greedy when it comes to glucose. Brain cells don't come cheap in terms of metabolism. About 20% of your resting metabolic rate is taken up by nerve cells in your brain. This relatively small organ takes up this huge amount of energy. And it's because neurons do all that metabolic work that you learned about last semester, with all those channels of opening and closing and getting the potassium and sodium membrane potential right and all that. So nerve tissue has to have glucose to live. It's the only fuel it can use. So one of the things that the body always has to do is maintain a blood glucose level. In other words, keep glucose in the blood so that the brain and other nervous tissue can continue to function. If we take the glucose out of a patient, first we get seizures, then we get a coma, and then we get a dead patient. So glucose is a necessity. And that pathway can happen within 20 minutes of a bad blood glucose fall. You can get seizure, coma, on the way to death. So glucose level is one of the most time-sensitive of all of the nutrient pool that we talked about last time. So I like this picture because it shows how each cell in our body, okay, so this is supposed to be like a representative cell, can actually do some pretty remarkable biochemistry that's very, very useful. Okay, so here's that nutrient pool that I keep talking about. So fatty acids, glucose, and amino acids. And we have anabolism is in blue, catabolism is in red. So amino acids form together to make proteins, right? That's the most complex structure amino acids form is proteins. They can be elaborate, thousands and thousands of amino acids long. Glucose molecules link together to form glycogen. Remember, glycogen is that branching starch, so that huge numbers of glucose molecules can be stored in a kind of granule, in a compact form, by linking them all together. I think we talked about that in a different chapter. So that we call that glycogenesis, is the formation of glycogen from glucose. All the bolded words here are important. Okay, so I'm going to try to get all of them. So when we go from glucose to glycogen, we call that glycogenesis. Glycogen can also be broken back down into glucose. It's like a battery. You can charge it up, and then you can release that energy. And when we break it down, we call that glycogenolysis. Okay, lysis is to break down. Genesis is to create. Okay, so you kind of get the words there. This is glycogenesis, the creation of glycogen. Glycogenolysis is the breakdown, the lysis of glycogen to make glucose. Okay, so we got our two sides there. We call amino acid to protein. This you talked about last semester. This is protein synthesis. That big elaborate process with the mRNAs and the tRNAs and all that stuff. Okay, that's this. Protein to amino acid, we just call that proteinolysis, but they left that out of this one. Okay, and then down here on the fat side, fatty acids are formed into triglycerides, and triglycerides are broken back down into fatty acids. This top column, these are structural. In other words, cells are made of this stuff. Okay, and then this is what comes into cells that cells use to make themselves and repair themselves. The whole bottom of this now is we can take these nutrients and we can feed them down into the mitochondria to produce ATP, as long as we have oxygen. So fatty acids can be broken down into 3 carbon chains and 2 carbon chains, which then get fed into the mitochondria. Mitochondria are picky eaters. Okay, they can't eat big molecules. They can only eat small molecules. So in order to feed the mitochondria and get ATP out, all of the nutrients have to be broken down into these 2 and 3 carbon chains, so little pieces. Okay, so we can do that 2 and 3 carbon chains with fatty acids. We can do that with glucose. We take glucose and break it, that's glycolysis, and we get 3 carbon chains, which become 2 carbon chains, which the mitochondria can actually eat. All right? Now we can reverse these processes too. We can create fatty acids by taking small carbon chains and building them into these long chains of carbon and hydrogen. We'll talk a little bit about that later. We can build glucose as well. So one of the ways we can turn fat into sugar is we take the fat, we break it down into 3 carbon chains, and then we take those 3 carbon chains, we stick them back together, and we make glucose out of it. Kind of a neat trick. We turn one thing into another. We call that gluconeogenesis. gluco for glucose, and then new formation. Neo is new. Genesis is to create. So we're making new glucose, gluconeogenesis. The liver does a lot of that. And then glycolysis is the breakdown of glucose into these 2 and 3 carbon chains. So a lot of interesting chemistry here. And so we can go from here all the way up. We can go from here all the way down. What we can't do, and the reason that we need plants in our lives, is we can't turn carbon dioxide into 2 and 3 carbon chains. That you need a plant friend for, right? And some photosynthesis to make that happen. So once you get down to the mitochondria, none of the rest of that is reversible. So the mitochondria can't work in reverse and create 2 and 3 carbon chains. That would be cool, because we'd be totally self-sufficient then, but we don't have that. We need our plant friends instead. All right, so a lot of flexibility. Okay, some questions, which once again, I forgot my phone. I've been terrible about that this semester. Okay, do it by hand. Cells synthesize new organic components. Why? We know cells are busy making things all the time, but which of those things is the reason? I'm not on soccer, folks. I'm sorry. So what do you think? Yep, all the above. So secretion requires synthesis, and that's one that students often miss is the secrete part. So like saliva, for example, you think, well, it's just water. What do we have to make? Well, in order for saliva to have its characteristics of being slippery and works to stick things together, there has to be something in that water. What's in that water is proteins called glycoproteins that have to be made in order to be put into the saliva to make that happen. So secretion always requires synthesis. And then structural maintenance, just like the things in our macroscopic world, things fall apart. Cell machinery gets used, and as a thing gets used, eventually it wears out and has to be replaced. So we have maintenance and then growth and repair, which is happening all the time too. All right, a little review from last semester with Sean. The carbon dioxide of respiration is formed at which point in that, what happens in the mitochondria? Well, is it A? Who thinks it's A? Nobody thinks it's A. Is it B? Who thinks it's B? A couple people think it's B. How about, is it E? Electron transport. No, you're not sure. You don't remember. Right. Okay, so the carbon dioxide in respiration is formed in the citric acid cycle. Here, let me back up to show you this. Interesting things happen in the mitochondria. It's one of the most, in my opinion, it's very metabolically complex in there. And it's one of these systems where if you were going to redesign life on Earth, you would probably make this a lot simpler because there's a lot of these different steps that intersect. But one of the take-home messages is the CO2 is made in the citric acid cycle, but the oxygen is used in the electron transport system. So there's a separation. We think, well, oxygen goes in and CO2 comes out. But in the mitochondria, it's not that simple. First, the CO2 is made, and then the oxygen is used, and the oxygen becomes water, not CO2. So where are those spots? Here's one. Here's, every time this turns, that's one cycle. So we get a CO2 produced here and a CO2 produced here, because we only started with 3, right? So the CO2 is produced in the citric acid cycle, but then the oxygen is used. Where's oxygen coming in? It's somewhere here. Here it is. Yeah, the oxygen comes in, the 2 electrons go out, and this is where the oxygen is used, is at the end, and it's actually used to make water, not CO2, which is a little counterintuitive to how people usually think. So do review those parts. I'm really not going to go over it because it's not very high yield. The citric acid cycle and the electron transport system, It's sort of like the cranial nerves. It's like a rite of passage in medical education. Everybody learns this very complicated system, and in real taking care of patients, you never use that information. Now, that doesn't mean you're not going to have to know it, because it gets asked on board exams and stuff, because they want to know that you know the biochemistry. But in taking care of patients, do you scratch your head and say, I wonder if there's something wrong with their Krebs cycle? Never. Never, ever, ever. So it's one of those you just have to learn it because somebody expects you to. Okay, this is a good one. What percent of energy released from catabolism is lost as heat? That was in an early slide that we talked about. Here, let's go back and find it. That's a little bit of trivia. It's right here. 60%. Right? See that right there? Now, that is what makes us exothermic creatures. You know, we produce excess heat, so we're warmer than our surroundings all the time. And the reason for that is it has to do with the efficiency of the electron transport system. If that system was more efficient, there would be less loss of heat, but then we would have a cold problem. You know, we would be cold all the time. Our body temperatures would be high. Now, we all know there are cold-blooded creatures. That doesn't mean that they don't release 60% of their ATP as heat either, or 2, because they do. You know, like snake, it has the same metabolism. It's just it doesn't try to keep its temperature at a particular spot. It just allows its temperature to change with the environment. You know, so they are. It's not that they're more efficient, it's that they use less energy, so they produce less heat, and they don't produce enough heat to keep themselves warm. All right. Okay. So that's metabolism in a nutshell, right? That's the framework of it. Now we're going to go back to the digestive tract, and we're going to see how those nutrient pool molecules actually get into the blood where they're used by the body. Okay, so by way of review, you know, the processes of digestion start in the mouth, and the mouth does have some actual digestion that occurs. Remember, digestion is a chemical process. There are enzymes in the mouth that digest starches, called amylases. The enzyme that breaks down starch is called amylase. And there's a lipase that comes from the tongue, too. So we're going to talk about those. In the stomach, we have a lot of protein digestion that occurs. In the duodenum, that's where we get the secretions of the liver and the pancreas that get added to the mix that are going to trigger digestion. In the jejunum, we get the final, where we get nutrient absorption as well as the last steps of digestion occur there. And eventually we get into the large intestine where we have indigestible material that feeds bacteria that live there. The water is removed from that. We call that compaction. And then eventually the indigestible material is removed or is expelled from the body as feces. All right. And then we'll talk a little bit about how these nutrients are used. Okay, so we do carbohydrates, proteins, and then fats. So we'll start with carbohydrates. I'm going to turn this light off. That's better. Okay, so carbohydrates found in nature are typically of the starch variety. In other words, long chains of simple sugars. And you all have had some chemistry, so you know what sugar is, right? You know, you've got a 6-carbon sugar, 5-carbon sugar, a lot of carbon, oxygen, a lot of oxygen and carbon in sugars. Well, all a starch is you take a simple sugar like glucose and you run long strings of them together and you have starch. Starches are typically what plants make as their own form of energy storage and to create their structure so that they can grow taller and gather more light and such. So the carbohydrates in our diet are mostly starches. In our modern American diet, there are some disaccharides and monosaccharides too. You know, so sucrose, table sugar, is an example of a disaccharide. Now, do you find that in nature? Sure you do. but in very small amounts. You find it in nectar and stuff like that. But we take that and we refine it. So we have some simpler sugars in our diet too. But what's interesting is only the smallest molecule, only the smallest building blocks can actually be absorbed into our own bloodstream for use by our own cells. So in order to use these carbohydrates in our diet, we have to take these long branching chains of starches and break them all the way down into simple sugars so that they can be absorbed. And it's a multi-step process, right? So carbohydrates go in, right? That's ingestion. This breakdown begins in the mouth. Salivary amylase, the word amylase means carbohydrate digester. So this is a protein that's going to break up carbohydrates into their smaller building block pieces. Salivary amylase takes starch and breaks it into disaccharides and trisaccharides. Now you can experience this with a simple experiment. If you take something that has a lot of starch in it, so a cracker works very well for this, and you just put it on your tongue, you do not chew, you do nothing but sit there and wait, eventually the cracker disappears. Well, what happened to it? Well, it got digested. The starches turned in. Starches usually have some rigid structure to them. But when you take that starch and you turn it into disaccharides and trisaccharides, it liquefies, right? So that's an example of salivary amylase in action. And so that begins carbohydrate digestion. So then we swallow, and in what we swallow, there's now a mixture of the food and the salivary amylase. So both go into the stomach. Salivary amylase continues to do its job in the stomach until the pH in the stomach gets too low. Remember, proteins are pH sensitive. So when you change the pH of a protein, it usually stops working. We call that denaturing. Okay, so salivary amylase denatures at about 4.5. So that means it's active for an hour or two after a meal. There is no carbohydrate digestion in the stomach except for what is happening by salivary amylase. So even though the stomach is a very busy place, it is lousy at carbohydrate digestion because it doesn't have any enzymes for it. The stomach's enzymes are all geared towards protein digestion. So once the salivary amylase kicks off at 4.5, a pH of 4.5 because of denaturing, that process of digestion pauses or halts. Okay. Eventually, what's in the stomach gets shuttled into the duodenum, and once that occurs, now the pancreas adds its amylase, which is pancreatic alpha amylase. And the process that salivary amylase started then continues in the rest of the small intestine, but by a different enzyme. Okay, so we get alpha amylase doing its thing. Now, the amylases can't finish the job. They go from long branching chains, starches, to disaccharides and trisaccharides. But all that can be absorbed are monosaccharides, single sugars. Right? So just, if your chemistry is a little rusty, okay, this is an example sugar, right? So that's glucose. If we string it to another glucose molecule, that's a disaccharide, right? Because there's two. And then if we add a third one over here, now it's a trisaccharide. Okay? So in carbohydrate digestion, we go from, I'm going to make a key here. Okay, so this is a sugar, and I'm going to draw it as a circle, right? So a starch looks like this. So it's these long branching chains of sugars, okay? So in this first step, the amylase takes this starch and it breaks it into, here's a chain of 3, here's a chain of 2, right? Here's a chain of 2, so chains of 2 and 3, di and trisaccharides. But we need to get back to simple sugars to be absorbed. So that last step, which is a common theme across all of these nutrient types, the last step of digestion, where we go from larger molecule to tiniest building block molecule, happens at the edge of the epithelial cells that line the small intestine. Okay, remember, call them Bart-Simpson cells because they have fuzzy edges, and as they line the villi, remember that? And I called it the brush border because that's what we call it, the brush border. Well, it's here at the brush border of those cells that the last step of carbohydrate digestion happens. So there's enzymes attached to the Bart-Simpson hair here, the microvilli is the technical name for that. And those enzymes transform disaccharides into simple sugars. So there's 3 of them. There's a maltase, a sucrase, and a lactase. So maltose is 2 glucoses. Sucrose is a glucose and a fructose. And lactose, which is milk sugar, is a glucose and a galactose. So there are other types, but those are the most common ones. These A's means to break. So these are enzymes that break these particular sugars, right? So maltase breaks maltose, sucrase breaks sucrose, and so on. And they live right here on the brush border. So the disaccharides come this way, they get broken down into the last step, right here, and then they're immediately absorbed into this cell as simple sugars, which then go right on through and into the bloodstream where they go out to the rest of the body then. You know, so the pasta that you had for lunch followed this path, right? The digestion of those starches started in the mouth, continued in the stomach until the pH got too low and turned that enzyme off. Then the pancreas added its enzyme, alpha amylase, which broke the starch down into disaccharides and trisaccharides. And then right here on your brush border, we get to simple sugars, and those simple sugars then enter the bloodstream, right? So in a meal that's high in carbohydrates, the blood leaving the digestive tract is very high in simple sugars. You know, very high in glucose, very high in fructose, and those other simple sugars. So when people talk about their blood sugar, they're talking about the blood sugar circulating to all the cells, not the blood sugar that's coming from the digestive tract to the liver. Because oftentimes the blood glucose there is very high. Okay, so just as a reminder, we start at the heart, blood goes to the digestive tract. Remember, the tract is the tube. From the digestive tract, it goes to the liver, and then it goes back to the heart, where then it can go out to the rest of the body. This is the key part of this, okay? All blood from the digestive tract goes through the liver before it goes anywhere else. We call this system the hepatic portal system. Okay, because it's a routing of blood. And it means that the liver can work on the blood that's coming from the digestive tract before it goes anywhere else. So the reason I bring this up here is, you know, you've had pasta for a meal. Well, the blood coming from the digestive tract is going to be very high in glucose, huge high glucose level. But as that glucose goes into the liver binds that glucose up into glycogen so that by the time the blood leaves the liver, the glucose level is normal for the rest of the body. Okay, so that's just one example of how the liver does its thing of regulating our metabolism and our nutrient levels in the rest of the body. Okay, so we have simple sugars coming in. Well, what happens then? Well, those simple sugars go to the liver, like I was just talking about. And in the liver, we have options. Depends on what the body needs. Okay, so if the body needs to store glucose, then the liver will produce glycogen. It will store the glucose. That's this way. That's going from these molecules to the original long branching chain, right? So that's one thing, is the glycogen may get stored. The glycogen, so the liver makes glycogen. Skeletal muscle also makes glycogen as well, and that's so that it has plenty of glucose around for anaerobic metabolism, think we talked a little bit about that last semester. But ultimately, glucose is either going to be used by the cells of the body or used only by the brain cells. Okay? Brain cells have to have glucose all the time. That's their only option. So they have no choice. The rest of the cells of the body, though, can either use glucose as a fuel for metabolism or can use something else instead. Insulin is that key that determines whether cells are going to use glucose or whether cells are going to use some other fuel. Because on all cells of the body except brain cells, they have glucose gates, glucose transporters, that have an insulin receptor that's part of them. So they only pump glucose in when insulin is present. And as we talked about in the endocrine system, that means when insulin is present in the body, glucose levels fall, right? Because the glucose is getting pumped out of the blood and into the cells where it's being used. So here, for example, the insulin is present, glucose is being pumped in. We create an ATP when we break glucose into 2 3-carbon sugars. Those 3-carbon sugars or 3-carbon fragments become 2-carbon fragments. We go into the mitochondria, magic happens, CO2 comes out, and oxygen gets used and ATP gets used. Right? That's the citric acid cycle again. So this is one, resting place. Where does the glucose that comes in go? Well, this is one place. It becomes CO2 in cells that have insulin present on their membrane, and glucose is allowed in to create fuel. Another place glucose might go is to form glycogen in skeletal muscle. A third place is to form glycogen in the liver. All right, so if glucose is going, did you talk about glycolysis last semester? I bet you did. Where you break glucose into 2 3-carbon fragments, feed it to the mitochondria in the first place, right? The words are different, but I'll give you the short version here, okay? Glycolysis is, okay, the use of glucose in the body. Of all of the metabolism of the mammal, it's like one of the things that is known the most about. Now, does that mean that glycolysis is like critical, super important in the body? Well, in this case, yes, it is. Glycolysis, we wouldn't be able to survive as the organism we are without it. But it has what I consider it's a knowledge bias. In other words, because we know so much about this process, there's a tendency to teach a lot about this process. I don't do that because it's low yield again. You're never going to look at a patient and say, oh, you have a problem with step 3 of your glycolysis cascade. No way, right? Because if they did, they'd be dead. They would never have been born in the first place because it's just that critical. So what I want you to know are the yields, the summary part. Okay, so not all of these steps. You will never in my class write fructose 1,6 bisphosphate, right? Maybe in your organic chemistry class, but not in my class, all right? So I just want you to know the concepts here. So the steps, or really not even the steps are that important. I want you to know what the yields are. So glycolysis, first of all, it takes place in the cytoplasm, not in the mitochondria. So we all think of mitochondria as the energy place. Glycolysis happens outside of mitochondria. That's an important feature, so I want you to know that. And it only yields 2 ATP. That isn't very much. So for a single molecule of glucose, we get 2 ATP as a net gain. The benefit, the reason why glycolysis sometimes becomes important is because it doesn't require oxygen to work. So you can actually do glycolysis even in a cell that's oxygen starved. Like you have in an anaerobic contracting muscle, for example. Okay, so I've been very specific in this slide set about what I want you to know. Know that a 6-carbon ring is broken into 2 3-carbon fragments that then get fed into the mitochondria, right? Okay. And some of my own personal bias shows through in some of this stuff. I hated this stuff in medical school and college, all the biochemistry stuff. So now I'm teaching it, so I get to teach not as much. So you get the benefit of that. And here we have more, this is what I call metabolic magic, right? So here's what we just talked about. Glucose gets broken into 2-3 carbon fragments. We get 2 ATPs out of that. Then we go into the mitochondria, and the wizard level magic happens, right? We've got all kinds of letters floating around here. We've got NADs, FADs, all kinds of this stuff. Well, in addition to what I showed you in the first slide where I wanted you to know the NAD and FAD and the H's moving around, here I just want you to know this part, that it results in 36 ATP being produced from every molecule of glucose. Now, why is that important? Well, number one, it makes it seem like you know more about the system than you actually do. You know that 36 is an important number. And it can fool people into thinking that perhaps you know more of this wizard magic in here than you actually do. But it also shows that what takes place in the mitochondria is very effective at producing ATP, right? A single molecule of glucose, and we take in a bazillion of these molecules every day, the starches that we eat, creates 36 ATP. So how do we fuel our hungry muscle when it's contracting and blowing through ATP in that sliding filament mechanism? Because the mitochondria are really darn good at making ATP out of carbon fragments as long as there's oxygen around, right? Okay. Here, we only have just a couple of minutes. I'll come back to that because I'm seeing a lot of sort of blank looks about the citric acid cycle and the oxidative phosphorylation. Okay, so we talked about this a little bit already. The trick with this is follow the NADs and FADs around, because that's where the energy is actually being produced. If this were a water wheel that's turning, turning a mill, for example, it's the NADs and FADs that are going to carry that energy. So that's the key. So over here I say, that we get 6 NAD molecules, 2 here, 2 here, and 2 here that are charged up. And we get an FAD molecule that's charged up here. Those charged molecules are then what power this part of the system. So those charged up NADs and FADs, they come in over here and they give up their electrons, and as those electrons, the charge, falls down this series of proteins being pulled by this hydrogen ion, this turns the crank. This system right here creates the energy that then shoots this hydrogen ion through this transporter, and this is what makes ATP in the end, right here. You see this spot right here? That's the magic end. All of this work in this system and in this system, it's all about this line right here. We power up or we charge up this system so that these hydrogen ions are flung out of this protein, and that passage causes a P to be added to an ADP to make an ATP right here. Now, where do those flinging hydrogen ions go? Well, they get bound up to an oxygen radical, right? That's an O negative. You never see those in nature except for brief amounts of time because they're highly reactive. But this O negative sitting here is pulling on this H plus even as this is pushing on it. So we get a lot of energy in this step, and we get a water made. Well, that's boring. But we also get an ATP made, a high energy bond that we can use for a bunch of other things. Okay. All right, so I'll talk to you a little bit more about why glucose as opposed to other things when we get to next time. All right. For